Thank you. Thank you, and many thanks first for the organizer actually to have me around. This is my first time in South Korea, a long travel, and bear with me if I'm not in full shape. So what I would like to share with you, I had this first slide actually, is to share with you what happened over the COVID-19, what was existing already, and how this translates currently to better preparedness. And this is just to indicate that I have some obligation with the Canadian government actually that might, I don't want it to be in any way a conflicting situation. Similarly, I'm very involved in discussion actually with the pastor network. And additionally, because there is some relation to my activity with the CEPI, where I'm on the Scientific Advisory Board. And so what I am presenting are my views and what has been done in my lab. Obviously, I'm a little bit influenced by the discussion, obviously with the high-level discussion with regard to pandemic preparedness. So what I'm showing here, and I definitely will not go on detail, is over the COVID-19, what happens is a real-time competition between the platform technologies to generate eventually vaccine. And what you see listed here are the different vaccine, obviously viral vector, mRNA. Everyone believes that this emerged, but it has over a long period of time of development, and we shouldn't diminish the work that has been done before. Recombinant protein, traditional vaccines such as inactivated and live attenuated vaccine. All this went into a kind of competition, which at different level, but the key result that came out with regard to addressing the COVID-19 situation was availability of vaccine, okay? And that's the primary one listed in this graph indicate, in spite of the challenges, the discussion that went over the adenovirus as a viral vector, that was the one that saved eventually most of the lives, actually. Combined, obviously, the mRNA was definitely present through the Pfizer, BioNTech, and Moderna. And other adenovirus, I think that you will recognize a number of these, Sputnik, Johnson & Johnson. Bharat came within very short timeline, actually, with inactivated virus vaccines, as well as Corona vac, which was also an inactivated virus vaccine. So the bottom line, and this is the lesson actually, I repeat, that was on real time. So the lesson was what to do with the knowledge that was accumulated over that period of time and what went on a daily basis. Actually, the diagram that I was showing from WHO was updated and showing how the progress was made. So the key question is, what should we retain for being better ready for the next pandemic? And we call it X. It's a disease X. It's undefined. So referring to the CEPI 100-day mission that emerged from, in fact, a recommendation that was from the G7, or G7 Secretariat, indicating that with what we have done with the COVID-19, we should go and be prepared, be ready to have a vaccine, indication of a vaccine available within 100 days mission. I'm not sure that everyone is familiar, but this is a driver now for the mission of CEPI. So this is taking into consideration what happened over the COVID-19, the different steps actually to come with the development of the vaccine that came on board, dominantly the mRNA and the viral vector, and from that, try to translate this to that targets aim of 100 days, okay? And I invite you, this is taken directly from the document of CEPI. So we need to have a safe, effective, and globally accessible vaccine within 100 days. So this seems just incredible, but that was kind of the critical demonstration of the COVID-19 that yes, there might be element to achieve this, but the solution is about being ready prior to that 100 days. The paradigm shift is to put in place the technology, the different step, the platform. This is the key point. It took a number of days, in fact, to clarify this notion of platform. For some of you, it's very clear, but it's a critical message, actually, with regard. So what's precede the 100-day is extremely important. This is where the main investment need to happen, and this is where the agencies are going on. In Canada, we have the HERC, Health Emergency Readiness Canada. Here in Europe, I'm pretty sure that you have a similar organization within South Korea. So platform technology for rapid response, I mean, based on what we just presented, came out as the mRNA with the demonstration through Moderna and Pfizer, viral vector not to be put aside in spite of the debate. I was working over the last 35, 40 years actually on the adenovirus, and it was highly valuable to see this technology emerging actually as, but there is other viral vector, and I will elaborate in fact of the potential. So the proposal is, and this is shared at high level actually, to work on other viral vectors such as the VSV, vesicular stomatitis virus, and the mRNA to advance and be ready with this technology platform. So I will present, and this is based on prior work that we have done, trying to support this technology because it has been already used for vaccination with the Ebola. VSV is a negative single-stranded RNA enveloped differently from the adenovirus, a quite large size, and the viral is through budding. So when we say a platform, what we need to take into consideration is the different step of manufacturing, essentially, and that's the key point. We are considering the manufacturing, including all the elements associated to design, and we will have a talk actually that will go in deep in the design of the vaccine using artificial intelligence. But the other step are as critical production, purification, analytical technologies are absolutely critical. Let's relate them to the regulatory approval and eventually address the problematic of thermostability or stability at large. So this is the current process flow done at Merck, in fact, to generate the VSV vaccine that has been used in the crisis of the Ebola. Working, not ideal. This is because it has been, this was originally developed by Public Health Agency of Canada and has been used as is. Critical is the development of the analytical tools. And when we started looking at this system, we had to develop the novel analytical tool, essentially looking at the infectious unit versus the total particle. This has to be validated. So through the assay, I will not go again in the detail. It's just to underline the critical elements that are required actually to get acceptance from the regulatory body when you go through this. Similarly, you have to validate a number of, and I am listing just two here, and there is a number of others that are associated to the critical quality attribute of a vaccine that is based on VSV. Critical input in the process is viral cells that are the substrate to produce the viral vector are adherent cells. So what we have done, looking extensively, we had to do de novo the sequencing of the viral cells. We adapted these cells to suspension. This means that you can scale up right away, actually, to produce extremely large quantities. It took a while, actually, but ultimately we were able to do it. We were very probably the first lab that successfully did it in serum-free medium, and that was demonstrated. So this is published for anyone who is interested by the data, showing this suspension achievement. So in the process, that's critical. It will align with simplifying the overall approach. So basic experiment with the Ebola vaccine that was available and with the construct that was available at that time. And we increased significantly the production. Scale up, which is part of the demonstrating this platform. And extremely interesting result, again, fully published. And so demonstrating that with one construct, with the COVID spike, we were able to generate significant yield from suspension culture, actually. And with ratio total to infectious, this is a critical quality attribute of this kind of vaccine, quite significant. This vaccine is temperature sensitive. I think it went to phase 1, and the COVID was controlled after that. In terms of, because this symposium is about innovation, one, with regard to cell culture production, one dominant approach is to use what we call the continuous manufacturing. Continuous manufacturing integrated between the upstream and downstream, using here 3 columns. This is to speed up and to reduce the volume that will be required to generate a certain quantity. So some work has been done on this that was, again, published, increasing the potential. And if there is something to refer to, we talk about densification. In fact, with rather small scale, within reduced timeline, you can provide the same quantity. And this is what is shown in a graphic here. Instead of repeated run, if you operate in continuous, you will shorten the time to generate a million of vaccine doses. So this is ready to be integrated to the platform currently that exists with the VSV. Critical with regard to defining a platform, equitable distribution came as a critical element in the discussion of the WHO treaty. Okay, and if there has been something observed with the VSV already, with the Ebola, that was distributed in Western Africa at -60. This is problematic. So the mRNA was also another problematic raised at -80 storage. So this is the reminder that when we develop a vaccine, and now it's becoming a target profile of the product that would be approved by the WHO for the pre-qualification, no extreme low temperature should be required for the product. So toward that end, and that was work which was ongoing, we did the demonstration of having formulation adequate for stability of the VSV vaccine, even beyond freeze drying was completed. That was building on work that was done with the adenovirus work. But again, this is an innovation that currently will be integrated, and that's a strong recommendation to the vaccine using the VSV as a delivery vector. mRNA, I will spend a little bit more time, less time, because this is a well-known platform building on 50 years of work. In fact, working with my colleagues at McGill University, there is a lot of science behind, actually, on that stability aspect that has been put forward over the years, the design, but it's still ongoing, actually. Significant improvements are yet expected. But the critical step are the sequencing identification. Recall that when we refer to X pandemic, it needs to be sequence agnostic. That's the case of the VSV. And it is the case with the adenovirus. That's why it's one of the preferred platforms. Then you need to deliver this mRNA, and with the encapsulation step, this is where there has been some knowledge accumulated over many, many years with regard, in fact, to trying to deliver DNA. Okay, the lipid nanoparticle didn't have not emerged at the time where the mRNA was developed. In fact, it was preceding for many 30, 40 years of research trying to deliver DNA. So importantly, the mRNA, not only the mRNA, the RNA technology has a broad scope of application. It can be technology that can be used quite effectively in cell and gene therapy, and treatment of a number of other diseases. We focus on the mRNA with regard to vaccination, but there is significant advantage actually with regard to the technology. And this is an invitation actually to the Institut Pasteur and the network. We are in discussion currently with Rebecca Gray, but also Institut Pasteur of Dakar, because we had at McGill University significant funding from the government of Canada over 7 years with Canada First Research Fund, which is labeled DNA to RNA. Without getting into the detail, actually, we cover all the aspect of translation from the discovery to the getting forward with product of interest to the patient. And there is an international component that currently is in place. So going to the manufacturing process, not to be underestimated, is the first step, in fact, the template DNA design and amplification. So often for the amplification, this is what has been done over the COVID-19. We refer to established technologies such as E. coli fermentation, but we are looking at the technologies that should be more friendly, more easy. This is the place to use synthetic biology at large, defining the different steps. It seems straightforward, and from someone having done a lot of cell culture, I mean, the mRNA technology is definitely more straightforward. The different steps are extremely well defined, not to under-diminish the challenges that might be posed, and I will go over. Critically, the critical quality attributes are once again something extremely important to implement right away, to follow up and to operate the process, and to guarantee the quality, not only the quantity, the quality of the product. So as you see, it's using quite well-established, analytical tools. This has to be implemented, but it's rather evolving. Currently, we do have a project, for example, that attempt to replace many of the very complicated techniques, mass spectroscopy, by a genomic approach, which is more informative using the nanopore system, for example. So the aim to solve the problem of generating, amplifying rapidly template DNA is to move to cell-free DNA synthesis. Okay, the system currently, the process manufacturing mRNA, use already in vitro transcription. So not going back to the E. coli fermentation, not because it's complicated, because building new capacity, you need that to invest on that part, actually, and the new investment are directed toward low-income and medium-income countries, actually. So the cell-free system is something that will solve this problematic. This is, in a nutshell, what we selected as a technology, which is called RCA, rolling circle amplification, and demonstrates significant potential, actually, to be further optimized. Using essentially an enzyme that is the Phe29, and there is already some optimization on the type of enzyme to get higher yield. We are talking about an amplification by a factor up to 4,000. The mRNA production can also be significantly improved by, I described the principle of continuous manufacturing. Similarly, here we are working on making the production continuous. This will enable having lower volume of operation and a densification of the process using an approach that is called intensification. Okay, these are wording that have been used more and more by process development experts. Right away, this is an accessible technology, actually, and we had a chance to discuss it we are in that space of synthetic biology, in fact. Almost everything is defined, and the optimization is very possible to do even better. So just running over some basic results, referring to the continuous manufacturing. When we think about actually the template DNA and the enzyme are catalysts in this biochemical reaction, meaning that they are not consumed. So that idea of recycling is dominant, and this is where the field is going. In fact, this will significantly reduce the costs. Okay, we were facing a problematic with regard to making the technology very user-friendly and accessible to low and medium income countries. So you still need to deliver that mRNA. LNPs are, and I mentioned that a long investment of research has been done prior to that in the cell and gene therapy field in the past with regard to DNA. Need to consider the critical quality attribute. This has to do with the target profile of the products, the size, and obviously, ideally, you should have a very reduced distribution. These are the lipids that have been extensively used, and as you see, there is a lot of potential, in fact, of significant improvement. This has been locked, but the field is open for targeting specific tissues through variation. The novelty is that is within that ionizable lipids, and significant work is ongoing in that space. Stability challenge, it's addressable in a nutshell. Everyone was aware, actually. I think that hospital for the first time discovered what was -18. In research lab, that was current, actually, but the reality brought by the mRNA was requiring -80. So there is a potential, and through basic work we have done, actually, there is capacity to get to, through addition of excipient, not only variation with the lipid component, but through excipient based on what we know from basic other biologics product, recombinant protein, viral vector. These can be used to design liquid formulation that are more controlling the stability and even freeze drying. So microfluidic encapsulation, this is the process by which, in fact, you encapsulate the mRNA within the the LNPs. This is, again, a very active area that is moving forward, and it's definitely achievable. We're talking about making emulsion, but in the most reproducible way. So, just listing some interesting results, again, with the liquid formulation and the freeze drying. This work is continuously published to demonstrate, in fact, the feasibility. In conclusion, we have two processes that adhere to the requirement of a platform as recommended by WHO and CEPI. The viral vector, and I gave the example of the VSV. Obviously, adenovirus remain also a viral vector technology, and mRNA technology. Why two? Because of the unexpected disease, it's very appropriate to have more than one technology in hand. So this work was over a number of years, actually funded by different organizations through, I mean, a very active team. And I don't want to end without inviting you to a conference that is of high interest in Porto. This is the 20th. This goes 20 years back. The value of this conference, it's a non-profit, is that it's a conference that put together industry and academia, and obviously researchers from the different fields. With this, I thank you all, and if we have time for questions, happy to take it.
Okay, thank you very much for your wonderful talk. I think we may have time for one or two questions. Oh, yeah, Professor Ian.
Given the problems with ITT, with the AstraZeneca adenovirus vaccine for COVID, do you think that adeno is really a viable alternative moving forward for disease X? And on the use of VSV, can that be used again, or is that a one-off type vaccine because of immune responses to the vector?
Your point is absolutely valid and reflects the deep discussion that took on at the CEPI meetings. Adenovirus, unfortunately, had a bad publicity, and I told you that I'm on that space over 40 years, actually. It went through the different challenges, and it demonstrated, and that's the figure that was shown, actually, that still it delivered billions of doses. And there is a huge set of data that have been generated. But currently, it's clear, the message is clear. We had some comment from Japan, that it's extremely badly seen, okay? So that's the proposal of looking to other alternative, a viral vector. When you think about the platform that are proposed, actually, it's delivering the signal and letting the body generate the antigen as a response, actually. I call the mRNA as a synthetic virus versus, the delivery with the VSV. So the VSV has that demonstration through the Ebola, because one of the conditions of being considered as a platform, it needs to have at least one product approved, okay? So it's a candidate that is second in the line. It's not a simple answer to your, but adenovirus delivered. And with the different serotypes that have been used, because when we go over the different manufacturers, different serotype have been used, actually. But I know that this is open for debate, and because we have to take into consideration that acceptance by the public, that's for sure.
Okay, yeah, next question.
Is it on? Yeah. So I was just thinking that one of the critical quality attributes of a virus a virus vectored vaccine is to remove host cell proteins. And in the case of the ChAdOx and the J&J vaccines for COVID, it may have been a adenoviral host cell protein that was actually contaminating the preparation that caused the binding to platelet factor 4 and caused the ITT. So if you can remove that from the mix, then perhaps you can get back to producing adenoviral vector vaccines, right?
You are absolutely right, actually. This is a field, actually, that when you deal with viruses, in fact, I'm part of the primary observation, actually. We had a discussion with WHO at the very first event that happened with the adenovirus. That was in gene therapy space, actually, the death of Jesse Kelsinger. And after that, there has been the HIV. But the reality, you are absolutely right, actually, that we were dealing with old process with not that much advanced technology, whereas the technology advanced, purification is absolutely critical, okay? And this might apply even more to envelope the virus with contamination of extravesicular material, etc. So yes, it's a point of attention where there is a need to make sure, actually, and that's where these critical quality attributes are extremely important, actually.